Okay. Thank you, Marina, very much for that introduction. She's always a bit too kind. Yeah, no, I mean, usually people, when they give a lecture, they start off by introducing themselves, but like today, the material is really interesting. So I think that we'll just start with that as fast as possible. And so I'm going to start with a summary or like discussion of the paper that you guys had to do the assignment for. And then we'll go into the topic of the day, which is representational drift. And we'll see, we'll try and finish as much as we can. I'll try and prioritize, not rushing. So we'll see what we can do. And yeah, so for this paper, this is a pretty good paper. It's really, really dense. So I actually took this course in undergrad, and like I remember thinking that each one of these figures is basically an entire PhD by itself, so like 1, 2, and 3. So this is a pretty like incredible paper, and to publish a really big paper in neuroscience, there's like generally two ways you can do it. Either like you find something new, like you find place cells, or you take something that we kind of already know, and then you just show it really, really rigorously and really, really well. And that's basically what Tony Gao was really good at, Jocelyn Franklin really good at, and what they do in this paper. So here they just, tackle the systems consolidation of contextual fear memories completely using the principles of necessity and sufficiency that you guys talked about last time. So I'm just going to start off with giving this high level summary of what they found all in all, and then we'll go through figures 1 and 3, which you guys had to write about. Also, just giving like sort of overview. And as we go, you can ask any questions regarding the assignment, like if there's variables that you're curious about or you want to hear about certain confounds or something, we'll play it by ear. So um, the the paper basically highlighted the mechanism of this contextual fear memory, and the way this goes is like starting with uh, yeah, there we go. So starting with the encoding process, what we find is that when a memory is first encoded, information about the context sort of comes in through the sensory information, through the sensory nerves, and it goes to the hippocampus. And then information about the shock goes to the amygdala. And the co-localization of those two things creates a sort of association that's formed between the two. Now, one of the main findings that they found and highlighted in figure 1 is that in this process of encoding, the medial entorhinal the medial entorhinal cortex sends signals to the prefrontal cortex that tag what is going to be a prefrontal engram that is going to support this memory in the prefrontal cortex. So during the encoding, these are selected. They're activated by the hippocampal cells, but they are not yet consolidated. And the way that they know this is that when they look at recent recall, they found that the medial prefrontal cortex was not necessary for the recall, whereas the entorhinal cortex was. Now, importantly, even during recent recall, the engram in the prefrontal cortex still exists. It's still there, living in the prefrontal cortex. It's been tagged during the encoding, and that's kind of what they show in figure 1. It's just that it hasn't yet consolidated sufficiently. It hasn't yet grown enough spines or gotten sufficient potentiation to just be activated endogenously by contextual stimuli. But the hippocampus is, and that's sort of what I show in this quote over here, is that the hippocampus has synapses that can potentiate extremely quickly, and that allows it to sort of encode everything that happens in an episode the moment that it happens. But the prefrontal cortex, in part due to its role in flexible behavior in the cortex in general, it does not stabilize very quickly, and that helps it stay flexible. But the issue is that memories will then not form very quickly. And what you guys talked about in previous lectures is that that's fine because over the course of a week or so, during the night and during rest, the hippocampus will actually undergo these sort of sharp wave replays, sharp wave ripples, and during which it will replay sequences of events that the animal has experienced. And while the prefrontal cortex stabilizes less than the hippocampus, it will eventually stabilize if you keep repeating something to it. And so that's basically what the hippocampus does. It will continue to replay and repeat and train the prefrontal engram to become consolidated enough to activate at remote contexts. And therefore, so at remote contexts, what you find is that the context now comes in and then goes to the prefrontal cortex, where memories are organized sort of semantically, and then it's retrieved through the connections between the prefrontal cortex and the basolateral amygdala. Here, the hippocampus endogenously becomes not involved in the memory, but again, the engram still exists in the hippocampus. And they show this in figure 3 when they activate it with blue light and find that freezing does increase. So again, the engram still exists, it just goes into dormancy. And this is relevant because now the hippocampus is freed up to then encode the next set of episodic stimuli. So this is the process of system consolidation or complementary systems. You have one system that can stabilize quickly, but then it has to keep moving on every month or so. And you have a system that doesn't stabilize very quickly, but because of that, it can stay flexible and it can be trained by like the hippocampus with replay and whatnot. So that's basically the summary of everything that they showed. It's, like I said, it was pretty well done. And now going over the figures that you guys had to sort of read and analyze in your assignment. So like I said, I'm just going to go very high level overview over what they found here. So I'm not going to go into details about the optogenetics or the chemogenetics or anything like that. But if anyone has any questions, please feel free. But I find it's helpful to just like give a very like simple summary because these figures are pretty heavy. So for example, all this top like quadrant can just be summarized by saying that here they just confirm that projections are sent from the entorhinal cortex to the relevant downstream regions. And then they just show that inhibiting this during training will impair the remote recall. So this is basically during the encoding process is when the entorhinal cortex will tag what will be the prefrontal engram. And inhibiting that, the sort of projections will inhibit that tagging process. And the same is not true for the anterior cingulate gyrus, the cortex, right? As for the rest of figure 1, they found, okay, that it's necessary. And so they next wanted to just see observationally, will we find this prefrontal engram is active during the encoding process. And so what they find here is just that the contextual fear memory will activate the prefrontal engram, and it's dependent on the entorhinal cortex, because if you inhibit those projections to the prefrontal cortex, then you won't see increased CFOS activity. And remember that CFOS is an immediate early gene that will tag activation. So in your assignment, when you guys are, Marina was telling me when you're putting like dependent variables and independent variables and such, she wants to stress that you want to highlight what is the relevance of an independent or dependent variable. So when you say like blue light is on or blue light is off, you want to say, okay, and this is the activation of channel rhodopsin opsins versus no activation. Or like when you say something like CFOS, you want to say this is operationalizing activity, in the hour or 2 hours preceding perfusion. So that's just like a comment regarding some of the submissions that were put forward so far. And here they now that they already showed the necessity in the earlier, they're here showing the sufficiency of the prefrontal engram. And here you can see that in day 2, when it is activated, even though it was not involved endogenously at recent recall, activating the prefrontal engram at recent recall will still cause increased freezing. Again, just highlighting the difference between like having an engram allocated and tagged versus having it already like consolidated sufficiently. And this last part is sort of linked to the top part, where they found that the prefrontal engram is downstream of these MEC neurons. So in the top part, they just found that the MEC was necessary But in the bottom part, because they used retrograde tracing that was CFOS dependent, they were specifically only retrograde tracing the prefrontal engram, and still found that that had sort of upstream neurons in the entorhinal cortex. So some confounds that you guys could like have mentioned, just to give you some ideas, is this. For example, this TET on, TET off system, where you're this doxycycline based system that allows you to measure the timing of the encoding. It necessitates the animals being on antibiotics for basically the entirety of their involvement in the experiment, except for that one day where you do the tagging. And so there are some links there where like chronic antibiotic treatment can impair microglia, can impair the gut microbiome. Like back when I was doing undergrad in HMB430, they talked a lot about this. So this is sort of one potential confound. Another issue with CFOS tagging is that even though we know that cells that are active tend to express CFOS, there are also cells that activate that aren't tagged by CFOS. So it's not necessarily that good always as an activity dependent marker. And this can this biases the engram field a lot, actually, because engrams are always involved, like by definition, they're defined almost by this sort of IEG activation, which is not always reliable. Yeah, if there's any question about figure 1, but I think everything is good. Okay, chat is good. Yeah, so figure 3, like I was saying, all of these are, fairly dense, but in figure 3, they really highlight the role of the hippocampus here, and the role of it sort of in the consolidation process and its role in the engram itself. So in this first half, they just sort of validate this method that they use where they selectively express toxins to kill only the engram cells that are, they express both CFOS and they did so during the encoding process. Here they're just basically validating it. And then they're showing that if you do kill the cells in the dentate gyrus, you're going to result in the lack of the formation of the prefrontal engram. So this sort of contributes to the part where we say that replay and this sort of offline replay is going to help the consolidate them over the week in between the recent and the remote recall. And that this sort of lack of consolidation process, you can see it in the spines, as well as in the activation of the actual cells recorded by fiber photometry. And for the last part of figure 3, they attack the dentate gyrus engram specifically. So first they just try and do an observational result to see like what's the sort of overlap of CFOS representations between encoding and sort of various points of retrieval. And they found that the dentate gyrus engram is only naturally active during recent recall. And the reason for this is because at remote time points, it's not very well consolidated. It lacks spines, basically the same story. But importantly, and I highlighted this already with the prefrontal cortex, if you activate it during remote recall, it's still going to increase freezing behavior. So just because the engram isn't consolidated naturally doesn't mean that it's not still there, in the sense that it still is connected to fear representing memories in the BLA. And you can rescue those connections by just activating enough of that population. And so this is basically the summary of this paper. Like I said, it was a pretty good paper, pretty dense, a little tough to read. And that's basically it for this part. But I'm just gonna open the floor again just for like 5 seconds. I don't like awkward silence very much, so I move on pretty quick. Okay, good. All right. Okay. Then I'll just like move on to my part of the lecture and see if this track. So today I'm going to be talking about something called representational drift, and this is actually a very recent finding. So this is called trends in neuroscience for a reason. We're basically like at the frontier of learning and memory research at this point. I mean, this is a phenomenon that was first ever observed in 2013, and it was still quite like contentious back then. Now everyone agrees that it happens, but there's still a lot of disagreement about why and whatnot. And yeah, you see a lot of the papers I cite are 2022, 2023. This is, pretty debatable still as to what exactly is going on. And the reason is that it flies in the face of so much of what we thought that we knew before. Because everything that you guys learned so far in terms of memory, for example, when you took place cells, you guys highlighted the stability of the place cell as a necessary feature of defining it as a place cell, that it will have the same place field over prolonged periods of times. And same for other types of cells like sound cells or time cells or whatnot. And when we talked about the engram, we also talked about stability. The idea goes all the way back to the beginning of the 20th century, like before all these molecular techniques. Richard Simon just theoretically pontificated that a memory must necessarily be some engraved version of the physiological process that occurred during the encoding. Like that's where the name engram comes from, the root of engraving. And basically representational drift showed that that's wrong, that representations actually change and are much less stable than we thought. And you can generally find some cells that are very stable for a long period of time, but those are the exception, not the rule. And yeah, so that's the quote won't make very much sense now, but hopefully it'll make sense later on. To stay the same, everything changes. And yeah, so I kind of already went through that first half. So today's content, I'm just going to start with what was it. So start with that first 2013 paper that first ever found it. And then I'll sort of organize the literature based on 3 different perspectives. Like I said, it's still quite messy. There's no like good consensus. But towards the end, I'll try and like put it all together, all the perspectives and everything. And we'll see how much we can finish, like I said, but at least I know we should be able to cover the first part. So this observation of representational drift came as a consequence of our improved technology in neuroscience, basically like every finding in neuroscience. So back in the day, in order to record neurons, people used, types of electrodes like tetrodes and whatnot. And generally those could record a couple neurons, if you're lucky, like a few dozen neurons. But nowadays we have things like calcium imaging, where that has sort of, you can record so many cells and it has really good spatial resolution, but pretty poor temporal resolution. But then you also have like incredible improvements in the electrode field, where you have things like neuropixel probes now that can also record hundreds to thousands of neurons. So nowadays, a single PhD student in a single day of research can record more neurons than his professor did in his entire career. And so it makes for a lot more things to analyze, but it also gave us a much greater perspective. And just as this technology was facilitated, we also found that we could record the same neurons for very long periods of time and actually maintain identity of which neuron is which. So this technology also facilitated having really long recordings. Back in the day, like you find the place cell, that's good, but you're going to lose it in a couple of days because the electrode keeps moving. And so you couldn't do these long form types of studies. And when we finally could, we got excited, and so we did just that. We implanted, or Ziv et al. Implanted, they sort of use this calcium imaging basically, where they can measure the activity of hundreds and thousands of cells in the dorsal hippocampus, and they just picked a very simple spatial paradigm, the linear track, which you guys have come across a couple of times. This is the first time we do something this long, so we thought let's just keep it super simple, barely even train the animal, just see what happens to place cells over really long periods of time. So there were no real manipulations, just imaging for over a month. And first they confirmed everything that we know so far. So on each day they were able to find place cells that tiled the entire linear tract. And but here, importantly, very importantly, in this figure, each day the cells are ordered based on their centroid location on that specific day. So here we're not looking, when we're looking across the same row, we're not looking at the same cell. So basically what I'm trying to say is this result found that each day had place fields that tiled the whole track, but they weren't always necessarily the same place fields. At least that's what we're going to see now. But each day had enough place fields for that day. And they sort of confirmed the characteristics of those place fields. For example, they always found that the beginning and end of the track was overrepresented, and it was similarly overrepresented on every day of recording. And they also found that most place fields had a direction bias, so they only activated when coming from the right or from the left. But next, what they did is they ordered all the place fields based on one day, and then they just looked at those exact same cells on each recording session. And what they found was surprising. So when ordering only so these are the place fields as organized by their centroid location on day 5. And you see here that while only 5 days later, there is a nice tiling of the track, these place cells basically stop representing the linear track. They just, most of them either leave, so they stop being place fields at all. Some new place fields jump in, and some also remap. But rarely do they remap. They generally just jump in or jump out. And you see this on every single day. So you can organize place fields by that day, and you'll always find a very clean sort of tiling of place fields on that day. But you check on close days, you find less of them. Check on farther days, less of them. Keeps getting less and less, which sort of flies in the face of what we knew. We defined place fields as being stable. Here they're stable within a session, but less so between sessions. And this is sort of problematic because we assume that the representation of space is static. So it's not like new rules are being learned. The representation is static, but the cells are learning. And it's kind of paradoxical to all our hypotheses about sort of memory, that stable representation will create stable behavior. But what about less passive memories, right? So that one was just our first time trying to image for long periods of time. And like I said, back then it was still a bit controversial that drift is happening. But people thought, what about sort of the representation of a task where the animal has to continue performing that task well to get rewards, or is punished if it doesn't perform well? Then the animal has to maintain stable behavior. Maybe that will stabilize the representation. Here, and also here they imaged the posterior parietal cortex, not the hippocampus. And they did so. This is basically like a choice task where you have to pick go right or go left. And well, they basically replicated the same thing. So even in choice tasks, in more abstract regions of the brain, you tend to look for sequences of cells that tile up the task space. So you guys kind of talked about this like a lecture or two ago about how we can have spaces that are more abstract than just the sort of three-dimensional physical world. So, and in those spaces, you still want to tile the entire sort of quote-unquote space of that task. And here they do. You have cells that tile from the sort of nose poke all the way to the reward, basically. But they also drift, and they drift in a manner that is sensitive to time. Let me check something. Yeah. And this is even though that the posterior parietal cortex is necessary. That is necessary. That's what the figure F here shows. is that when you inactivate the posterior parietal cortex, the animal can't perform the task. So we know that these representations are needed for good performance. And we also know that the mouse continues to perform very well for the whole 30 days. So behavior is stable, but the representation isn't. And that's weird. And that's, yeah, summarized over here. So representations keep learning even when behavior isn't. It's stable generally on the order of days, but not for very long periods of time. But importantly, it's stable even when the animal isn't learning anything new. And it challenges the classical theory of learning, which is that stable behaviors are created by stable representations. So that's basically what drift is. It's in the name. We have cells that the particular population activity should represent the task, and yet those tend to drift, to change, and it tends to be with time. And since Ziv, like I alluded to, this has been replicated many times now. So, and it's been replicated all over the place. Generally, in many areas that we see, that we can image, we find it in the hippocampus, in the piriform cortex, which is a sort of olfactory association cortex, and even in the visual cortex, which was surprising to some people, because you'd think those are like V1 we're talking, so the first visual cortex. So representations should be tethered to their sort of external sensory input in some way, and yet they still drift. But it's controversial still in motor areas. So some places show that they see it, some show that they don't. Some famous papers have shown that, like in bird songs that are very stereotyped, there is 0-0 drift. So we've seen it in motor areas. You can be tethered to the output, but in sensory areas we still see a decent amount of drift. And also we tend to see it whether we're doing electrophysiology or calcium imaging. So it's a pretty robust finding, even though it's a little bit still disagreeable. And there are some characteristics of representational drift that we seem to find hold, despite which region you're looking at. So for one, it's primarily a function of time. Frequency of exposure to the relevant variable can slow it down. In one paper, they found that for the olfactory representations, if you keep giving it that same smell every single day, that representation of that smell will drift more slowly. But generally, time seems to be the most important factor. So there are other studies in the hippocampus where they vary how many times they show the animal the maze, and it doesn't seem to affect drift rates as much. So it's a bit contentious. But time is the most important factor. At least everyone agrees on that. People disagree of whether it's the only one. This is from the Ziv et al. Paper. You can see that they calculated the probability of a place cell coming back once it had shown activity. And you can see that, from day 1 to day 2, there's always a sharp drop. Like that first day, a bunch of the place cells you reported are gone. But then after that, it tends to be pretty linear with time. And this was also, generally replicated in many places. But also this posterior parietal cortex paper that I sort of talked about last part also found that it's tended to be linear with time. The other thing that I sort of alluded to already is that while some neurons can remap, generally drift is just like whole new like subpopulations of place cells or engram cells or whatever you want jumping in, and a bunch of the ones that reported yesterday not activating. You see remapping, but it's just less common. Yeah, so in this sort of posterior parietal cortex paper, they just sort of show in this graphic here that you find new neurons, they point them as gain, or some leave, but you don't see as much rate remapping. And when you look at the place field changes, they tend to always like be very strongly centered at 0. Again, so listen, the place fields are not shifting, they're just disappearing or coming back. Shifting a little bit sometimes. And the last sort of really important characteristic is that the majority of cells that take part in representation do it for that day. We find them for one day, and they tend to be good place cells, and then they disappear. That's kind of what you can see here. But if you look, there are cells that are active for the entire month. So if you were Moser or an older neuroscientist back in the day hunting for place cells, you can find place cells that are stable for 30 days. This is just importantly that when we image many cells, we're able to tell what's the exception and what's not. And we find that stability is not an exception, even though the percentage of cells active in the whole population stays roughly the same at like 35%. That's what the inset shows. So no more or less cells are being engaged. And so that's the representational drift. I will summarize it. So the classical model of memory says that at encoding, certain engrams or representations or place cells are tagged. They become a part of the representation. And then every time you go there, they become more and more consolidated because they keep being retrieved. So it's like, oh, they're useful. And eventually an expert is an expert because his engrams are so consolidated that you can guarantee the retrieval. But those are still the same cells from the first day. It's just that they kept getting stronger and stronger. And so that's what the classical model of memory here shows. Like when you see a lightning bolt, you just see that the cell was activated, versus the shading is supposed to show the sort of potentiation. So, and the degree of shading is supposed to show how potentiated they are. So here you can see every time acquisition day 1 and acquisition day 2 retrieval, you're activating the same cells. They're becoming more potentiated. They're reactivating next session again and again. But in the new model or this dynamical model that we're forced to come to with sort of this representational drift findings, is that yes, those first cells that were activated, they will consolidate, but on the next day, some of them just won't fire, and some whole new cells will fire. And so you're going to get some lightning bolts on the consolidated neurons and some on new place cells or new representation cells. And this happens every single time. Every time you encode or every time you retrieve, this happens. New cells jump in, new cells jump, or old cells jump out, and you just basically get this sort of representation where here the color represents which day they joined the memory, and it's more so like a mosaic of representations where the various tiles of that mosaic were encoded on different days. Some yesterday, some 2 days before, and most of the ones that will activate will be the ones from yesterday because drift is generally time dependent. So that's what drift is. And like I said many times now, it's kind of confusing because it's not very stable. But actually in other little subfields of neuroscience, it's not a new idea that you can have stable idea with unstable dynamics. So specifically when you look at like people who like to really zoom in deep when they're doing neuroscience and look at like dendrites and spines and things, then you see that dendrites are always coming in and out. And they do so pretty rapidly and pretty dynamically. So this idea that like a synapse is potentiated and it holds the memory, and let's say if 1 month later the animal is retrieving it, it's that same synapse that was potentiated. We already knew that can't happen because the hippocampus, every single spine turns over in 4 to 6 weeks. And so we just never took it to the cellular level. And when you zoom in even more, you find that like inside spines per se, in the molecular dynamics, there's always a turnover of things like AMPA receptors and whatnot. Like it's more of an equilibrium Some are always being exocytosed onto the membrane, or some are always being endocytosed. And that actually the system is in slight disequilibrium, which means that more will get eaten slowly with time. And so, there's no component of a memory that truly stays stable, and yet information can remain stable in the representations. And that's sort of what we want to try and get at. And people have been thinking about this for a long time. It's actually something fundamental about making systems that are continuously learning, because when we try and think about learning and memory, we try and think about them as a vacuum. This was acquisition day, this is where learning occurred, and this is retrieval day, this is where retrieval is occurring. But any day, as far as the brain knows, new rules can happen. And so every day is an encoding day and a retrieval day for the brain. And it's tough actually to make systems that can learn continuously. Like even in AI, where we have made models that can learn pretty well, we still can't make anything that can continuously learn. If you try and take something like ChatGPT and retrain it on a new task, it will undergo something called catastrophic forgetting. So it's tough to balance both. And that's kind of the theme of the whole lecture today. Like going back to 2005, there was this really influential theoretical paper that tackled this idea. And this quote is really good. It says the main challenge in building long-lasting memory, especially in cases where new experiences are continuously generating, is protecting the memory trace from the ravages of ongoing activity, not from the ravages of time. And this paper that I'm highlighting here was very influential. It talked about the role of epigenetics in this. So even if a neuron, doesn't have the spine anymore, it can have a memory of the fact that it had a strong spine with that neuron in its epigenetics. But that's not good enough for us, because we're still finding that the whole cells are changing. And so again, I got a bit ahead of myself, but this is a fundamental problem about learning memory. It's always happening. If you notice in the papers that we tend to talk about, we almost always talk about fear conditioning. But that's because fear conditioning is one of the rare memories that you can have extremely strong encoding in one day, and then just define retrieval really easily in later days. People don't like to do n-gram research on tasks where it takes weeks for the animals to get good at, because when did the n-gram happen? They want this very simple delineation between learning and memory. And that's why we tend to use the fear conditioning task, because you know that the animal can learn it very quickly. It's a very simple task. But most things in life are not like this. Most things are sort of continuously being refined, or you don't know if new rules are going to come up. Even when I'm retrieving something, I'm standing here and I'm retrieving to you guys all that I know about representational drift and n-grams and whatnot, I'm still experiencing something new at the same time. I have to encode this memory as well and learn it as well. And that balance is actually really hard to make. It's fundamentally a problem of having parameterized systems where you have memory spread over the weights, in the case of synapses, as we have here. Making sort of computers or computations that combine the processor and the memory component into one thing is actually super non-trivial of a reason why neural networks work. If you think early in the history of computation, when Turing wanted to make thinking machines and whatnot, early in the thinking of computations, people thought, okay, processing will happen and then memory will happen somewhere else. But the idea of having the same sort of thing that does the processing, which is the weight or the synapse, also store the memory, gives a lot of good mathematical properties to these networks that makes it good for learning. And it's good for memory, but it's impossible to solve the continuous learning problem so far as we know it. in like AI and now and stuff like that. Like it's still one of the largest unsolved problems in AI, the problem of catastrophic forgetting. So insofar as representational drift might tell us something about how brains continuously learn, it's really exciting field. So if you guys just remember that, I'll be happy. But next I'm going to be talking about the theoretical perspectives, but let's just do it 5-10 minute break. Um, yeah, we'll be back at 2. Yeah. Yeah. I think it's when you like train an AI to let's say do task A, and like let's say you train AI to associate context A with a fear. So and then it learns it. But then you train the AI to do associate context B with a shock. And you find it learns that, but then when you go to test the original one, you can't. Yeah, unless you like interleave the training, it doesn't like learn. Yeah. Something in the chat. So like the first here you can see that you compare the different games. So then you see it's like the five here and the three. So like farther the more you see no matter what is always after the media there's some it doesn't come slightly less short every day. So like 0 is like I mean 5 days, Yeah, this is like a program distribution, right? So there's self-efficient. Not like there's always that. Yeah. Hey, guys, good. Second. Okay, guys, I think I'll continue now. So, so this second half, like I was saying, is pretty still like a little bit contentious. What's happening? What's not happening? It's hard to sort of bring this together with the engram research because all this representational drift stuff, they do it with calcium imaging, where you're imaging the cells, but you're not doing like CFOS tagging. You can only do CFOS tagging once. So it's pretty tough to combine this with the engram stuff that like we talked about in the Tonigawa paper and things like that. And so that's been a challenge in sort of unifying this with our other like theories of memory. But we still have some pretty decent ideas, and so that's what we'll go through now. So the first perspective out of the three that I'll walk through today is inspired from the research in n-gram allocation. So first I will take a small like detour into this idea of n-gram allocation. So when you first ever learn a memory, then cells that are active in that moment will become the memory cells. But what dictates which cells are those? What gets allocated or becomes a part of the n-gram? And what research has found is that what determines which gets involved is this term that's a little bit hand wavy. We just call it excitability, which is how jumpy the neurons sort of are. Is that it's not always the same as like the synaptic weights. You can have sort of excitability changes. Like I don't know so much about in the pyramidal neurons, but at least in sensory neurons and stuff. For example, if they change their voltage gated sodium density in their axon hillock, right? That's a way to change excitability without affecting synaptic weights. So they're not always perfectly linked, and we find that this excitability is what sort of the cells that are the most excitable become a part of the memory n-gram. And once roughly 30% of the cell population is active, then interneurons in the population will sort of inhibit the rest of the population and make sure that only the most excitable 30% activated. And then those most excitable 30% will become a part of the n-gram. So here is just like a summary figure showing that the cells that just happened to have increased excitability at pre-training, then they became the cells that became part of the n-gram. A lot of work also used CREB. So we find that CREB, the transcription factor, is linked to this idea of excitability. Cells that are overexpressing CREB will also have a higher likelihood of becoming a part of the n-gram. And then there's also this term of lingering excitability, right? Because we notice that if you just have a bunch of cells and with my electrode, I stimulate like 3 or 4 of them, and then I train the animal, those neurons that I stimulated are going to be more likely to jump in to the n-gram. So there's this idea emerged of like lingering excitability, like they were just recently activated and they're still in a state of sort of hyper excitability. And recently this was even like replicated with optogenetics. So if you just randomly activate population in the amygdala or hippocampus or whatnot, and then you do conditioning without the blue light, without the activation, you're still going to find those cells become the n-gram cells. So this idea of lingering excitability naturally led to a hypothesis. It said, well, if the cells that were just active are excitable in the next time step, what is the n-gram of memories that are very close in time look like? What if you have 2 memories that are different, but you encoded them one after the other? And so that's the question that this paper tried to answer. Also a pretty influential paper, where they would sort of allow animals to learn about one context, and then they allow them to learn about another context, and they vary the time interval between those 2 contexts, so 5 hours or multiple days. And they found that memories that are close in time have a large n-gram overlap. So they actually share a lot of the same memory cells. And even if you fear condition one of these contexts and then measure freezing in the other context, you found that if they first learned about these close in time, the animal will actually show freezing to the context it never learned fear conditioning in. And it did that only because those contexts were encoded close in time. And because those are encoded close in time, they have overlapping engrams. And those engrams are what form a connection between the hippocampus and the basolateral amygdala to create the fear memory. That's what this sort of figure shows, that you can do the fear conditioning in this blue container, and you still get freezing in the red container because the red container was explored only very close in time to the blue one. And this has been sort of replicated a lot now, and even review papers are out and everything. So it's a pretty well established idea that memories close in time have overlapping engrams, and also memories that are medium distance apart actually have less likelihood of having the same engram cells. So this is a sort of pretty famous summary slide that goes into more detail than we need for this lecture, but it's a pretty good figure. You guys can have it in your notes. And it just shows that, in those 6 hours after a memory, there's a high chance of co-allocation of the same engram cells for different memory. But in long periods of time, they're actually sort of become, unbiased away from each other. Which already, you can look at this and start having some ideas about this representational drift. Like, okay, so what you're telling me is, one day later, the excitability is going to be quite different than it was in the first day. And so is that not going to affect the retrieval process? Is that not going to cause new engrams to be encoded? Possibly. So that's why there's ideas to be had from here. That what we get from here is that excitability changes in a manner that's related to time. And like I said earlier, it's both synaptic plasticity and excitability that affects which neurons activate. So if excitability is changing with time and representations are changing with time, maybe it's the same thing. So that's the first perspective, is that representations close in time will have similar engrams, and that this can cause drift at retrieval stages. But importantly, you'll also timestamp your memories. So when the brain is doing retrieval, when it sees two engrams that are close in connection, it will know that these are close in time. When it sees two memories not close, it will know that it's not close in time. So this idea of timestamping to create discrete episodes, it's important one, because even let's say paths that you walk every day, you still want to encode every day's walk, even though you're probably not going to retrieve it. And you're going to remember, oh, this is when I walked, on Monday, and it was a little different when I walked on Tuesday. So you want to keep re-encoding, even though you're seeing the exact same place. And there is a new variable, even if your behavior is stable. And that variable is time. Like the experience of an expert animal doing the same task the 100th time is slightly different than its experience in the second time. And maybe that's that sort of timestamping of the different experiences is kind of what we're seeing. Because every memory starts out as an episodic memory, so you just want to capture it all sort of indiscriminately at first. And basically, what this hypothesis would say is that these cells are not just coding the task; they're coding the task and day one. So of course, on day 10, the activation is different because we're not on day one anymore. And in support of this, the same author that did the original representational study, like the Ziv et al. 2013, he built an AI that would take in the engram activation of the hippocampus across the month, and it would try and predict what the day is. And he showed that this can successfully be trained. Like basically what he's showing is that there is information in the representations about which day it is, and an AI can find that information. So that's the first perspective, is that memories drift to timestamp them over different time periods. And it kind of naturally leads to the second perspective, but I already alluded to it a little bit, which is that not only is time changing, but the animal is most likely having slightly different experiences. Maybe on day 100 he was hungry before, you know. Motivation is different, emotion is different. Maybe he was just socially defeated by his mate in the cage an hour ago. Like like these affect the excitability of the neurons. These are going to affect which cells can activate in the retrieval time point, and these might also be, you want to still encode these as episodic memories, because maybe they're important. So that's what this second perspective says, is that maybe drift allows a sort of baseline malleability, and this allows you to encode novel variables, motivational, emotional, stuff like that. It allows you to potentially learn something new if the rules of the task change, which they might on any day. You never know which day they will. And even if the rules never changed, you will still sort of have this malleability that just emerges, as like leaving room for it to potentially change. So even when rules aren't changing, you're still going to see changing representations, because the brain is sort of waiting or looking for something to change. So it's like a baseline malleability in the representation, and it can also encode time, motivation, stuff like that. So this second perspective basically answers the learning memory dilemma, which we alluded to in the introduction. That's saying that you're trying to learn and remember at the same time. So how can you learn new associations in old contexts without the old association interfering? And they're saying that you keep changing the representation so that the old one doesn't interfere with the new one in case the rules do change. And if the rules never change, well, at least we have good timestamp memories now. So that's what I say in these paragraphs here, that if the task rule changes, the evolving population can encode them. If the task rule does not change, stable performance is still maintained as you pass on the information from group to group. So someone in the chat kind of talked a little bit about this. They said, is this related to the idea of the reconsolidation, where when you reactivate a memory trace, you potentially could change it? And yeah, this is sort of related to that idea where it's saying every time you retrieve, you're also transferring that information to a new sort of representation. It's related, but it's not the same as reconsolidation. Because importantly, like in reconsolidation, you're reconsolidating the same original trace. You take that original trace and you change it. Whereas here what we're saying is, no, the trace keeps getting passed on from population to population. The information is being passed on. And one paper actually found some sort of support for this idea. That original posterior parietal paper that we've talked about a little bit now, yeah, they imaged for 30 days and they found that representations drifted. But then on day 31, they tried a new association. So they changed the connection between, what stimuli tell you go left or right. And they then looked at the neurons. And what they found was that new neurons sort of entered the population for the new rule at a roughly similar dynamic to the way that neurons entered during the drifting process. Here they're showing them in the black cells. And also some of the cells that represented the old rule in day 30 are still now representing the new rule in day 31. So we're still going by this drift idea where the representation moves with time, and if something new is learned, it's passed on to the moving representation. And if something new is not learned, the representation still moves. But it's just that the fact that it moves allows for the possibility that you can learn something. And like I said, this is sort of different than reconsolidation, because we're not reconsolidating the original memory trace. We're sort of writing new ones with time. But think about it, like if you had a rule change that occurred very close in time, then you will be more so reconsolidating, like changing the original trace. But when you have sometimes like long periods of time, then like for example, when you do extinction for long fear memory or something like that, you find that a new memory is a new population. It's not the same sort of old population. So if rules change close in time, you want to still change that original representation. But if rules change very far in time, you still have that memory of like, oh, I remember when the rule used to be like that, and then it became like that. And this sort of drifting process would allow for that. Another thing that this sort of baseline malleability idea would allow is that it would allow for the combination of temporally distinct but related memories. So we talked about how, memories that are close in time will have overlapping memory engrams, and that's useful. But you also want memories that are far in time but very related to also have similar overlapping engrams. And just the first perspective doesn't give you that. If excitability is only changing as a function of time. But what this sort of perspective puts forward is that if you have memory A and memory B, and they were encoded at different times, so they have different engrams. When you link them, the process of you having that linking moment, whether it's happening during the replay offline or online, then you're going to transfer this sort of linking to this new population. And this new population will sort of contain information about the relationship between A and B. And in that case, you will have like a strong sort of overlap between the new representations for A and B that you just encoded now. And so this idea was discussed in a recent review paper just on theoretical ideas about drift. There is one paper in the motor cortex that seems to support this idea. So they recorded in the monkey neurons as the animal was doing a reaching task. What the task is doesn't matter, but they were reaching, and you could try and sort of see what's the engram for a reach at this angle, and what's an engram for a reach at that angle, and whatnot. And what they found is that representations drifted. In this paper, they found that representations drifted. So you might have like the first 3 neurons on day 1, and the neurons 4 to 6 on day 2, on day n, sorry. But when you plot the activation of those neurons, you find that the dynamics are incredibly similar. So what do I mean? What do I mean by plotting the activity? Over here, you can imagine that like if each axis is the firing rate of that neuron at a particular time point, then the line sort of denotes the change in the firing rates of all 3 neurons over time. And so get the laser pointer out. Yeah, where it starts over here is like, it means that N1 is firing at a high firing rate, but N2 and N3 less so. That's what defines the XYZ, is the firing rate of these 3 neurons. And then as this moves, it's just showing that as the animal reaches, this is how the firing rates change over time. They do this sort of arc. And but after a while, these neurons leave the representation, and you find new neurons that encode the representation now. But when you plot the dynamics of those new ones, you find that it's very similar. That the firing rate of the 3 neurons tend to follow like a similar dynamic. So what they're sort of suggesting is that even though the neurons change, they are still representing the same information, and they do so in the sort of dynamics of the way that they move. So if the reach involves, a sequence of motor activations, then the N1, N2, and 3 communicate to similar motor outputs as N4 and 5 and 6, such that the same sort of order and pattern of activation will create the same like reach response, even though these are like different neurons. So that's sort of what they found, is that in representational drift, you have uh, a lot of change in the individual level of the individual neurons, but when you plot the dynamics of the population, it's surprisingly stable. And this is actually like a very popular field now, like in theoretical and neuroscience, or like even in experimental, because we can record so many cells. We see that even though individual cells, they have a lot of variability and whatnot, the population tends to move in quite predictable ways. When especially when you plot it like this, like I don't know if you guys have experience with like principal component analysis and things like that, but when you plot sort of the population activity of 1,000 cells in 3 dimensions, you see like very stereotyped behavior even with drift. But that's something that's out of the scope of today. I probably shouldn't have gone on that tangent as much as I did, but yeah, this idea of like, the population remaining stable even though the neurons are changing, it's a very popular like mathematical side of representational drift that we won't go to. But this here, it's supporting that this idea that the same memory is being transferred from representation to from cell group to cell group. With each retrieval is a new encoding. uh, and then the last perspective is something that I will go over after a 5-minute break because I'm doing good on time. Yeah. All right, guys, I'll continue. Okay, so it's just this last perspective. So this is sort of inspired from the idea of the n-gram specificity hypothesis, which is also then inspired from the encoding specificity hypothesis. So the encoding specificity hypothesis is from psychology. It goes way back, like behavioral cognitive psychology, which is just this idea that you're most likely to retrieve a memory when conditions most closely resemble the conditions during encoding. The reason I put a picture of a scuba diver is because there's a very famous old psychology paper that wanted to like take this point to a silly level, basically. So it trained people to memorize a list of words when they were scuba diving and when they were not, and they showed that if at retrieval they were scuba diving again, they retrieve it better. And the same is actually true even for like drugs. Like if someone is drunk at time of encoding, so believe it or not, they'll do better retrieving if they're drunk at retrieval. So this is like a fairly like well characterized idea. The retrieval is best done when conditions closely resemble encoding. But the issue is that, you know, what if at retrieval the excitability pattern of the population is very different than when it was during encoding? Then how are you going to sort of reliably retrieve that memory? What I show sort of here is just that, like what we were showing earlier, that some cells have hyper-excitability, some cells also have hypo-excitability, and those are especially unlikely to join the engram or be activated in any way. And so, yeah, basically, if you're relying on one single memory trace, then it's a little bit dubious because the excitability state is very different later on. Maybe you will fail at retrieval, right? Like generally, when you're studying for something, let's say you might be happy when you're studying it for it, and then at the test you're gonna be like nervous, so you don't want this interference to corrupt the retrieval. So what this third perspective says is that you want to have multiple traces for the same memory, and then that way you can guarantee that at least one of those traces will match the excitability pattern at retrieval. So, you know, like a good memory is a memory that you can retrieve no matter what. You know, whether you're sad, happy, angry, you know, you never get that feeling where it's at the tip of your tongue. And so in that way, you want it actually to be buffered against differences at retrieval. And the classic theory doesn't like account for that as well. So I'll show a little cartoon just to animate the process. So if you know in the first day there's some stimulus, let's just think one task relevant stimulus only, and you have, you know, an ensemble that encodes it. Then the next day you present the same stimulus, and just by happenstance, the neurons that you used for encoding yesterday are hypoexcitable. They're sleeping. They're not going to be activated right now. And so what do you do? Well, you just encode the memory again in the most excitable batch that's left. You do as if it was encoding day, since this original association isn't being retrieved. And so now you have a new representation. But importantly, both representations encode the same memory. So at retrieval, if the first one is sleeping, you can rely on the second one. If the second one's sleeping, you can rely on the first one. And so the more and more you keep going to the same place, the more and more redundant representations of that same place you have. And that's why, you know, with more training, your retrieval will become guaranteed, is what this perspective is saying. It's not that the same trace keeps getting strengthened, but it's that now you have so many different traces for this memory, you know, you're good. Like no matter what day you go, you're good. One sort of important idea behind this is an idea that we discussed in the Tonigawa paper all the way at the beginning. If you remember, even when an engram is sleeping, it's still an engram. So the hippocampus representation, when its spines all went away and it stopped being endogenously activated at remote retrieval time points, still, if you shine blue light, you'll activate it. So you can have offline cells that are still engram cells. It's an important idea. There's these sleeping cells are still engrams, basically. And this cartoon just goes on to say, okay, in day 3 now you retrieve the second one, and then in day 9, let's say the second one's sleeping, so now you retrieve the first. So again, just that there's just this idea that you are increasing your redundancy. So like, and that way making yourself more robust to failure. Like it's a pretty common idea in neuroscience. Like in HMB430, we learned like, oh, you know, we have so many parallel circuits for the same computation. It's very, very common in neuroscience to sort of have redundancy and to make sure things don't fail. And whereas parallel circuits are redundant in space, this is arguing for redundancy in time. Yeah, basically I said all this already. And this is just, you know, sort of summarizing it so that as in more difficult tasks, as the performance improves, the classical theory will say it's because that original memory trace that got allocated during acquisition is getting stronger. But the drift-based hypothesis would say that performance improves because you keep adding more and more, and you're now sort of robust to changes in excitability. In other words, you have like a form of excitability buffer. So now, even though in the test I'm extremely nervous and I'm in a context that I've never been, and everything is, all these things are different, like my excitability state isn't so different from encoding that I can't find my memory trace anymore. So those are sort of 3 perspectives on possibly trying to explain why drift occurs, and this sort of all put together here. Again, just like going back to it, we went through it earlier, but now you can sort of gain an appreciation. At first, sort of the reason in the dynamic model that you don't always get the most potentiated neurons activated is because of different excitability patterns, and that these differences can encode both time, they can encode novel representations, they can transfer the representation from one to another, and they can also sort of create redundancy where you have more representations of the task than you can ever activate it at any one same time point, but you have them in the back pocket. And putting it all together, I put together one figure that kind of combines the ideas also, but I made this figure, so take it with a grain of salt. It's not from paper or anything like that. But the idea is that when you have a good like expert animal, like this is in an already expert animal, then in the top box, what I'm showing is, you know, a potential pool of engram cells, or you can think of them as sub-engrams if you think of like the chunked versions. And you have more of these than you can activate at any one time point in the expert. And then, you know, you get like a retrieval queue, and you suddenly have to activate a bunch. And that's what we show at the bottom here. Now you're activating. And how do you pick which engrams you're going to activate from the pool of possible activations? Based on the functional connectivity and the excitability. Eventually, once you have so many neurons that are active, interneurons that are sort of regulating the population will do negative feedback, basically. You know, having more than 30-40% of your neurons active in any one population is excitotoxic. It's very important to control, so it's pretty like well controlled. And yeah, this sort of summarizes the various different ideas. So, you know, one example I like from like early on that sort of compares the classical and the dynamical models. I'll like go over. It's a little bit of like a hand wavy sort of conceptual example, but it helped me wrap my head around it like early on. Is that if you think of like the classical model of learning, like trying to just like maintain one trace, it's like when you learn something new as a student and you have to like write an essay about it or give me a presentation about it, but maybe you didn't have too much time to prepare, so you read one review paper. So you have a very structured sort of argument that you can try and give me, and you can't, it's not flexible, you can't really deviate from that review paper too much. If you try and like give me, if I ask you certain questions, you're going to answer it and you're going to go through the sequence that you learned yesterday pretty faithfully. But let's say someone like me, I'm talking about representational drift to you guys right now. I read my first paper in like 2020 and I had an episodic memory of it then, and then I learned new things, and sort of every single time that I learned more, that was like a new episodic memory. I added new engrams, and those ensembles were different each time because I was reading different papers, I was in different time points, I was in different motivational factors, whatever. But there are also so much that links these things together. And eventually now when I'm standing here in front of you guys, I'm not just relying on like one sequence trace that I'm really just trying to faithfully follow, but rather every time I reach into my mind to get a fact, I'm picking up like this like one sub engram or sub ensemble, and like maybe I use as I talk to you, I use an ensemble I encoded a week ago, and then an ensemble I coded a year ago, and then it's sort of like this idea, like I was saying, it's like a mosaic tiling of an engram, where each of the different tiles come from like different days, and together they make like a mosaic memory. And so that's the idea of like when you have like when you have an expert retrieving to you, he's just retrieving things from different times and points and in different ways and doing so in the moment. And if I ask the same question to you the next day, maybe he'll retrieve them in different order or say the argument completely differently. Even though I asked him the same day, it's just, or today I called him a different excitability state, and now his argument is takes a different pattern, sort of, even though it's the same like core ideas. But yeah, so that's the sort of summary slide that puts it together. And very recently, there was a really good review paper that also sort of summarizes so much of what we saw. It was released this year, like a couple of months ago, from Sickens. So it was published in Neuroscience. So it's, like I said, we're really right at the sort of frontier. And they just basically imaged the hippocampus of mice as the mice were sort of doing a task, but importantly, the task had changing rules. So the rules would change, and they change either in a structured or unstructured way, but like we won't like get into that part of the paper. And then they just like imaged every single day. And so they imaged for like 19 different sessions. Okay. So at the top, you're seeing like the first session, the bottom, the last session. And they're just sort of counting how many neurons were involved in the ensemble on that day. But importantly, they're coloring it based on the first time they ever saw that neuron become a part of the representation. So that's why that first bar is entirely red, because that's the first session of recording. So every cell that they saw it for the first time in the first session. And so they're all red. In the second one, it's split sort of 50-50. So half the cells were from yesterday, half the cells we saw whole new encoding today. And you just keep going until the 19th session. And what I want to highlight is just sort of the various colorations of this. So you see that in the 19th session, they have some cells that were encoded in the first day, and then a whole bunch of the ones encoded all throughout the middle, and then some of the ones at the end. And this sort of supports this idea of having like mosaic ensembles, basically, that every day you're using, some from yesterday, some before that, some from before that. And there's generally like a primacy or a recency bias, which means, you know, the more like a lot of representations tends to happen to today and to the first time we ever encountered the task. Like you see the red is still quite prominent. And part of the reason why Ngram, like necessity specificity studies also still work. Like you know, that the cells you tag on that first acquisition, they are still pivotal a month later. Well, yeah, this was a really good paper. And that's, you know, sort of it for like the main ideas of today. I'll just like talk a little bit about like some future stuff. Maybe if you guys are, like I said, this is like frontier, you know, so I don't know if any of you guys actually want to become neuroscientists, but if you do, and you know, make one of these ideas your research question, then I expect to be on the paper. But yeah, so one like massively unanswered question is, how do neuromodulators affect nerve? I think that that's like still like gaping in the literature, and honestly should not be like that difficult to test. So someone needs to get on it. And then, you know, do different brain regions drift at different rates? Is a pretty important question also, because let's say drift is related to some cognitive factor, then you should see that like associative areas should drift faster. You know, prefrontal cortex maybe drifts faster, but like visual areas or motor areas, like the closer you are tethered to external input or output, if drift has some sort of cognitive role in learning and memory, maybe, you know, rates differ in a important way. Also, like maybe, you know, we know this memory involves, you know, the posterior parietal cortex and the anterior cingulate cortex. Okay, do the those regions co-drift during a task, you know, or do they like drift separately? Is also like a pretty important idea that will give us some ideas about, you know, what's going on. Like this sort of review paper recently said that, yeah, like uh, they should co-drift because of like recurrent feedback. So as the sensory area is drifting, you know, that'll create some error which will cause the association area to follow, and so on. But these are still untested. And yeah, if drift rates are the same everywhere, then maybe we say, okay, this is just a biological inevitability, just, you know, the dynamic nature of these biological systems and whatnot. And yeah, so that's future outlook. And then also something very important is all of these are observational, like pretty much, unlike the Ngram where we were talking about specificity, necessity, and these like really strong sort of logical correlates that we can do experiments on. Here we don't have a way to manipulate drift directly and like have it be an independent variable. So that's something that we wanna be working on. So excitingly, there is a paper that very recently was able to modulate drift with exercise. They found that if animals exercise, then their representations drift faster, and that also if animals exercise, it increases the neurogenesis and in the hippocampus. And so the authors of these, actually the authors of this is also from Sick Kids, also published this year. I met this guy Snoop, like he gave us poster talk last year, and it just published like 2 months ago. But the idea is that basically the new adult-born granular cells, as they integrate themselves into the new circuit, they increase drift. And because exercise increases adult neurogenesis, it seems to increase drift. So we know that exercise also increases things like neurotrophic factors, like brain-derived neurotrophic factor and stuff like that. And if you take like a petri dish of cells and you like give them brain-derived neurotrophic factor or stuff, you'll see their neurites and axons just start growing all over the place. So I think that there's a pretty good hypothesis you could make that neurotrophic factors, especially because they're involved in like growth and like changing synapses, might be involved. And so again, yeah, that's just like a free research question idea for you guys. And yeah, that's it for today. Thank you. I left my email, so it's in your notes. So if any of you guys want to reach out, if you have like any questions, grad related, representational drift related, general neuroscience related, you just want to come to Sick Kids and like meet the guys I talked about, then yeah, please reach out. And yeah, I hope this quote makes more sense now. Yeah, so I finished early, so that's good for everyone. Yeah, thank you all, and thank you, Marina, for the opportunity and just generally for like, teaching you guys this course. She's always been a great teacher in my experience. So yeah.
Well, thank you so much, Adel. Thank you for showing that everything I was teaching previously is completely wrong. Thank you for confusing my students, but hopefully you'll also know that science isn't easy, and this is a preview of how things work in neuroscience, because you start graduate school, you have this beautiful hypothesis, everything's going to work out, you just have to run a few of these experiments, and then you analyze the data and you realize that everything is wrong, and you're in tears because your expectations aren't met, your PI's expectations aren't met, and everything is wrong. But this is where all of the excitement lives. So thank you, Adel, again, for showing how exciting neuroscience is. When we learn that we finally know something, we find something else that shows that we're wrong. And this is how we keep moving, and this is how we keep getting better and smarter. Again, thank you, and we will start a new set of lectures starting next week. Anytime, please feel free to schedule a meeting with me, and if you have any lingering regrets regarding the assignment that you submitted today, again, feel free to express those, because you still have a chance to do really, really well on your final assignment, okay? Yes. Yes, I will be posting assignment 3 and assignment 4 simultaneously. Yes. So no, you will not have to review the material from the first half, but in a way, some stuff, like for example, place cells, right? They will appear again. So this kind of underlying knowledge should still be there, okay?